Hi, I'm Liz Davidson and I'm going to talk about my work on barcode reference databases for UK freshwater arthropod species. DNA-based identification can provide a much more efficient approach to the identification of specimens, but accurate identification is dependent on comprehensive and reliable barcode reference databases. The barcode coverage of monitored species in Europe is much higher for fish than invertebrates. 87.9% of fish species, but only 64.5% of invertebrate species. The Darwin Tree of Life project and the FreshBase project aim to create collections of high-quality sequences for UK species. Knowledge of where the current gaps are will enable sequencing of missing species to be prioritised. This study analyses the coverage and quality of barcodes for UK freshwater arthropods. Arthropod species from the FreshBase invertebrate list were cross-checked with the lists shown here and synonyms were recorded. The final list contained 3,251 freshwater arthropod species in 32 orders. Searches were conducted in bold and reports and sequences were downloaded and analysed to assess the coverage of UK freshwater arthropods and also the quality of the stored sequences. To summarise some of the results, the gap analysis showed that 73% of species have at least one barcode in bold, but in some groups a high percentage of barcodes are stored privately. 60% of species have at least one publicly stored barcode. Only 37% of species are represented by at least 5 public barcodes. Having multiple barcodes for each species is really important in being able to assess quality. To accurately identify UK specimens using DNA-based methods, it is important that there is a clear local barcoding gap between species. This is an example plot for UK Plecoptera, showing the intraspecific against the interspecific distances. The species in the bottom half don't show a local barcoding gap, which could lead to difficulty in identifying them accurately. But further analysis shows that the lack of local barcoding gap here is sometimes caused by errors or misidentifications in the reference databases. Analyses for each order showed that 19 out of the 32 orders include species that currently do not show a local barcoding gap. We also looked at the origin of the sequences stored in bold. Only 5% of species are represented by barcodes from specimens from the UK. With such low representation from UK specimens, it's important to understand if it matters. To look at this, we analysed the geographic variation within species. This principal coordinates analysis shows the similarity in barcodes for one of the UK Plecoptera species. The barcodes from UK specimens are mostly grouped closely together with those from Norway, France and Switzerland, but separately from sequences from Portugal and from those from Germany and Austria. This and the other species we've looked at suggests that high geographic variation within species could sometimes contribute towards a lack of local barcoding gap. Without sequences from UK specimens, we don't know how similar they are to those from other countries. To summarise, 60% of UK freshwater arthropod species have at least one public barcode in bold. Only 37% of species are represented by at least 5 public barcodes, which makes it difficult to assess intraspecific variation and evaluate the quality of stored sequences. 19 of the 32 orders include species that do not show a local barcoding gap, but some are caused by issues that can be resolved through curation of stored barcodes. Only 5% of species have sequences from UK specimens, which are needed to assess whether they are similar to sequences from other countries. Thanks to all my supervisors and also to Ben Price at the Natural History Museum. Thank you for listening.